my name is Osheen and I'm working as a research assistant in Science for Life Laboratory, Sweden. I'm delighted to be here to share with you all my small project on segmenting cancer epithelial using only the information of nuclei morphology. The main challenge of the project was to understand if it is feasible to detect the cancer cells in complex tissue sections by nuclear morphology. This would allow the use of the channel for other stains or antibody markers, as well as in future enable disease characterization directly from bright field images. The dataset which was used for the project consisted of immunofluorescence stained DAPI channel images and their corresponding panepithelial channel images. The total number of images were 374, and a manual quality control on each set of images were performed to ensure that there are no artifacts and the content in the image is clear enough so that it's easier to train the neural network. To accomplish the task of segmentation, the workflow was divided into 5 subparts, namely ground truth generation, image tiling, deep neural networks, hyperparameter settings, and results. Each of them I will be discussing in the following slides. So the first step towards the method pipeline was to generate the ground truths. As my data was missing the ground truths, I only had DAPI and panepithelial and no ground truths. I have used Elastic, which is open source software and is user-friendly. It's a supervised machine learning software in which the experts train the computer more interactively by marking or scribbling in the region of interests, as you can see here in the blue or in the yellow label. And the algorithm learns the pixel classes and performs the prediction. To the next step was to reduce the size of the high-resolution fluorescent images, and this was due to divide the images into small square patches called tiles. The main aim of this approach was to make the images small enough so that they can be used to train the neural network and will require less memory. Here, 2 different tiling sizes were used, 128 by 128 and 1025 by 1025. This was mainly done to decide which size is appropriate to train the neural network. And finally, all the experiments were conducted on 1025 by 1025 tile size, so that even the fine structures in the image are clear and it is easier for the network to learn the smaller and finer details in the image. Once all the pre-processing of the images were done, the third step was to decide on which neural network to train. And to begin with, the first choice was UNet. And due to obvious reasons, as it's easy to train and implement in Keras and has fast computational capability, and it also performs great in biomedical image segmentation tasks. Addition to the UNet, the residual unit was the second choice of neural network, where the skip connections in the network within the residual unit facilitates more information to propagate and perform better. And the third choice was VGG16 with the transfer learning approach, where the dense layer was replaced with the convolutional layer to adapt to the segmentation task, because VGG is actually fully connected neural network, and so all the dense layer were converted to the convolutional layers, because here my task is to perform segmentation. And before training, the last step was hyperparameter setting. So hyperparameter settings are the variables that govern the entire training process of the neural network. And hence different settings were performed, different hyperparameter settings were performed and tested to match the best combination, which resulted in highest Dataset score. And also cross-validation was implemented to compare the performance of the 3 neural networks and also to prevent the model from overfitting. After the training was done and the neural networks were evaluated, the learning curves for the respective network architecture looked somewhat like these. They were pretty promising. And now let's have a look on the training scores. So the training scores were recorded for all the 3 neural networks separately. And the highest score was recorded by residual unit on the test set for 90.07%. And that for the transfer learning was 86.49 on the test set. Also, the 3 neural networks unit, residual unit, and the transfer learning were compared using the 3K-fold cross-validation, as I mentioned in my previous slide. And the best performing model out of the 3 was residual unit with the 88.05 Dataset score and the standard deviation of 3.19. Here are some prediction results from the residual unit. So the first image is was the input image to the model, to the neural network models, and their corresponding ground truths, which were generated from the elastics. And these are the prediction, and this is the prediction from the residual unit model. Here is the second example. And as we see from these predictions, they are pretty much closer to the ground truth. And with this, we can conclude that the deep neural network have the capability to use the morphology information to detect cancer epithelial. And this allows us to use this channel of pan-epithelial for any other staining or antibody marker, which will provide with other relevant information and will help in microscopic image analysis. Also, I would like to add here is, as we see in this prediction, that there are some fine structures that are predicted by the model and are not present in the ground truth. So more work can be done on the ground truth generation part so that we can make our ground truth much better from the elastic, and the prediction can be even more appropriate in that way. So with that, thank you so much for your attention, and I'm very happy to take questions in the end.